Welcome back to the Sand Science Experience, where we explore the most exciting new research papers and preprints just for you. Before we dive into today's really mind-bending science, please do take a moment to like this video and subscribe to the channel. It truly helps support the Sand Science Experience and lets us keep bringing you these deep dives into cutting-edge research. Okay, so today we're tackling a really fascinating mystery. Why does human embryonic development take, about 60 days? While a mouse just speeds through it in 15. It's a huge difference.
It really is.
This difference in developmental pace, scientists call it allochrony. It's puzzled researchers for a long, long time. We know the sequence of events, the order things happen in development. That's often quite similar across species. But the speed, that varies wildly. So our mission today, here on the Sand Science Experience, is to unpack some groundbreaking new findings. They point towards a really fundamental cellular mechanism, protein degradation. So if it's not just like big versus small, what's really driving this difference in speed, even down at the cell level?
Well, that's what's so intriguing. The basic molecular machinery, the genes and pathways involved in development, they're largely the same, conserved between us and mice, for instance. Yet the tempo is just so different. A really key model system for looking at this is something called the segmentation clock.
Ah, yes, the segmentation clock, not a physical clock, right?
Exactly. It's this cycle, this oscillation of gene expression that basically lays down the body segments one by one during embryogenesis. And here's the kicker. The human segmentation clock takes about 5 hours to complete one cycle.
5 hours, okay.
Compare that to the mouse, its clock cycles every 2.5 hours. So twice as fast.
Right, so the human clock is literally running slower. Okay, but what's controlling the speed of that clock at the molecular level? That's where protein stability fits in, I guess.
Yeah.
I remember some earlier work hinted at one specific gene, HES7, may be degrading slower in human cells.
That's right, there were hints, but this new research, it just took a massive leap forward. They systematically compare the degradation rates, how fast proteins are broken down, for about 5,000 different proteins. They did this between human and mouse cells, specifically induced presomitic mesoderm cells, or iPSM. These are cells relevant to that segmentation process.
5,000 proteins. How did they even measure that?
They used a really powerful technique called dynamic SILAC-based proteomics. It's basically a sophisticated way to label proteins and track precisely how long they stick around in the cell before being degraded.
Okay, SILAC. Got it. And what did this massive comparison show?
Well, the main finding was striking. A pervasive trend of slower protein degradation in human cells. Just generally slower turnover.
Slower across the board.
Pretty much. Think of it like this. Human proteins, on average, hang around for much longer before the cell recycles them. The median half-life, the time it takes for half the amount of a protein to disappear, was 37 hours in the human iPSM cells.
37 hours.
Compare that to just 24 hours in the mouse iPSM cells.
Wow, that's more than 50% longer on average.
Exactly. And this wasn't just a few outliers. They looked at over 3,600 proteins found in both species. For over 1,750 of those, the degradation was significantly slower in human cells. Significantly faster. Only about 126 proteins.
That's a huge skew towards slowness in humans.
It is. And it was seen everywhere. Proteins in the mitochondria, the ER, the nucleus. And it involved both major breakdown systems in the cell.
Both the ubiquitin-proteasome system and autophagy?
Correct. The ubiquitin-proteasome system, which tags individual proteins for destruction, and the autophagy-lysosome pathway, which handles bulkier cleanup jobs. Both seem slower overall in the human cells.
Okay, that is a really fundamental difference.
Yeah.
But finding what is slower naturally leads to, well, why? Did they get any clues about the underlying cause?
Right, that's the crucial next step. And this leads to an important question.
Yeah.
Can we actually, change this developmental speed? We know human iPSM cells have a different metabolic profile than mouse ones. They tend to have lower rates of glycolysis sugar breakdown and also lower mitochondrial respiration, sort of a lower metabolic hum.
A slower metabolism.
In a sense, yes, at the cellular level. So the researchers decided to test this link directly. They took mouse iPSM cells and treated them with something called 2-deoxy-D-glucose, or 2DG. It's basically a drug that inhibits glycolysis.
So they deliberately slowed down the mouse cell's metabolism.
Precisely. And the results were remarkable. Treating the mouse cells with 2DG actually extended the period of their segmentation clock, made it run slower, closer to human timing.
Whoa, really? Just by tweaking metabolism?
Yeah. And crucially, this 2DG treatment also shifted the protein stability profile in the mouse cells. Remember, the untreated mouse median half-life was 24 hours. In the 2DG-treated mouse cells, it jumped to 36 hours.
36. That's incredibly close to the human 37 hours.
It's strikingly similar, yes. It seems like slowing down metabolism, specifically glycolysis in this experiment, pushed the mouse cells towards a more human-like state in terms of both clock speed and protein stability. Now, 2DG seemed to affect the proteasome pathway a bit more, but the overall results strongly suggest metabolism is a key knob controlling protein degradation rates and, consequently, developmental tempo.
So they didn't just find a correlation, they showed they could actually tune the tempo by fiddling with metabolism. If we're kind of running on a slower metabolic clock right from the start, cellularly speaking, well, it makes you wonder about the bigger picture, doesn't it? Could this be an early hint about why humans live so much longer than mice, for example? Is lifespan linked to this fundamental pace?
It certainly opens up that line of thinking. This systematic analysis, it doesn't just confirm this widespread, slower protein turnover in human development compared to mouse. It really highlights metabolism as a critical modulator, sort of master dial for setting that pace. And yes, it absolutely opens up new avenues for understanding how these fundamental cellular processes scale across different species.
Yeah.
It might influence how we think about lifespan, aging, not just the speed of early development.
Yeah, absolutely. You start thinking about species-specific disease progression, maybe even aging itself, a really deep dive into the fundamental pace of life. Well, that's all the time we have for this particular deep dive. Fascinating stuff. Please don't forget to like this video and subscribe to the channel. It really does help support the SANS Science Experience. We'll be back soon with more exciting research for you. Thanks for joining us on the SANS Science Experience.